So let's talk about what can actually, what different types of memory and on what can facilitate the formation of memory on the cellular level. So memory can be short-term and long-term. Well, as a matter of fact, you have some immediate memory once you receive that information in the cerebral cortex. And then some of that data is selected for the formation of short-term memory or working memory. Now, some of that short-term memory is forgotten, and that's important. So some of that data are lost permanently. But some of the data become long-term memory, and it's important to understand that for this, short-term memory, the transfer of short-term memory to long-term memory mostly is facilitated by excitement. So when you have very strong, when you have very strong emotional impact on you, it will be memorized better. The formation of long-term memory is facilitated by the rehearsal. When you repeat the same thing over and over again, you memorize it. And if you can associate some of your working short-term memory data with the stuff that you already know. So, for example, you go into Anatomy and Physiology 2 class and you start talking about the regulation of hormones, and the concept of negative feedback comes up, and you already learned it, and now you can associate the hormonal regulation with the previous knowledge. It's important to understand that some of the long-term memory can also become irretrievable. Okay, so you can forget this. Okay. And the amount of information that is stored at each step progressively decreases. Now, what happens at the cellular level? At the cellular level, memory is formed in a way by changing in the regulation of gene expression, epigenetically or through transcription factors. That change in transcription may result in the deposition of certain proteins at synapses, for instance, increased number of ion channels, which will alter synaptic plasticity and synaptic strength, so-called long-term potentiation. It may lead to actually changes in the number of synapses and shape, even shape of dendrites. And overall, the memory formation will be associated with the changes in the density of receptors and or ion channels, and it will be associated with the amount of neurotransmitter that's released from presynaptic neuron. So all those various factors, changes in the transcription, amount of proteins on the synapse, epigenetic regulation of gene expression, all of it contributes to the memory formation. Now, in terms of function, memory can be declarative and procedural. So the example of formation of declarative memory is here. The information is transferred to the associated prefrontal cortex and then to hippocampus. Okay, so hippocampus stores declarative memory such as names of the students in the class roster, or list of the body parts. And declarative memory can very easily be forgotten. Now, interestingly, in so-called procedural memory, the experiences, the memories that are formed are hard to be forgotten because those are pretty much motor skills. So in this case, the main structures that are involved in the formation of procedural memory, one way or another, are associated with the motor function of the brain. So you can see that those inputs that you receive are directed to basal nuclei, and then to premotor cortex. And you may remember we talked about it, that premotor cortex forms patterns of movements that are transmitted to primary motor cortex. So basically, procedural memory will be something of a sort like riding a bike or writing. You can hardly forget how to ride a bike or how to write, although if you don't do it very often, you may be a little rusty with your skills. Now let's talk about some of the brain trauma stuff. So we've got traumatic brain injury first. In traumatic brain injury, the basics is that brain just hits the skull. Here you have coup injury, and skull stops and brain keeps moving, and then it bounces back and creates a counter coup injury. The mildest traumatic brain injury is concussion. Concussions can have cumulative effects. The clinical signs and symptoms of concussion are headache, inability to focus your vision, nausea and vomiting, double vision, vertigo. So concussions accumulate, leading to the chronic traumatic brain injury, and the main complications is that certain parts of the brain stop functioning, basically. A result of the severe concussion is contusion, which is a bruising of the brain. And if there is an injury, if the injury is substantial enough, contusion may lead to a coma. Now, damage to the blood vessels that supply the brain can result in subdural or subarachnoid hemorrhage, and that's a problem because blood accumulates in subdural or subarachnoid space, increasing intracranial pressure. And since we're talking about adults, the skull cannot give in to the pressure, so compression, that increased pressure results in the compression of the brain. These hemorrhages have to be surgically removed, but if they are not timely removed, the injury results in inflammation, which leads to local edema, and brain edema can be actually a cause of death in many cases. Another pretty large type of the problems in the brain are cerebrovascular accidents or strokes. Two types: the ischemic stroke, shown here, and the blood clot. Blocks the blood flow through an artery, and here you can see atherosclerotic artery. The lack of blood supply results in the death of neurons, not only neurons, but neurons in particular, because they cannot perform anaerobic metabolism. And if we're talking about, say, motor cortex in a frontal lobe, then stroke in the left side will result in a paralysis on the right side, on the contralateral side. Can result in various sensory deficits, can result in the motor deficits like eye movements, inability to properly speak. Some people experience transient cerebrovascular attacks, transient strokes, and that is basically you have a clot that blocks the blood flow for a very short period of time. It still damages the neurons, but it does not affect them as tremendously as the actual full-blown stroke would. Usually there's transient ischemic attacks in the brain circulation are the sign of oncoming stroke. Hemorrhagic stroke is shown here. Aneurysm, that is usually the result of the constant elevated blood pressure, ruptures, the blood spills into the parenchyma of the brain, resulting in inflammation, edema, damage to the brain tissue, and so on and so forth. Now, 4 diseases of the brain that I would like you to generally know. So if we're going to start with Alzheimer's here. So in Alzheimer's disease, 2 main pathological structures are accumulating in the brain. Those are beta amyloid plaques formed by beta amyloid peptide, and neurofibrils tangle up axons, and these neurofibrils formed by tau protein. You can clearly see the even structural deterioration of the cortex and brain tissue in advanced Alzheimer's brain compared to the healthy brain. So usually that leads to dementia, loss of memory, loss of language, and ultimately death. Now here I have a little bit of a mislabeled. So Parkinson's disease is actually here. So that's Parkinson's. So you can see that the activity of basal nuclei in control compared to the symptomatic Parkinson's is drastically different. So controlled healthy brain basal nuclei are much more active than symptomatic Parkinson's. So the mechanism is quite elegant. Dopamine releasing neurons in a part of a midbrain, neurons in substantia nigra, they basically die and they cannot produce dopamine. The role of dopamine is to release the inhibition of motor pathways imposed on them by basal nuclei. So when no dopamine is present, inhibition persists, and that results in hypokinesia. The symptoms of Parkinson's disease: slow movements, stiff face, forward bent posture, so basically not enough movement. There are treatments, dopamine precursors, dopamine agonists, but it's a progressive neurodegenerative disease. Now in Huntington disease, here you can see the brains of a person with the Huntington's versus brain of a person without. The healthy subject can see slight ventriculomegaly, for instance, and slight deterioration of the size of the brain parenchyma. It results from the accumulation of the protein called huntingtin. And that accumulation of huntingtin, the mutant huntingtin, results in the degeneration of basal nuclei, so movement will be dramatically impaired. You can see that basal nuclei are much more smaller, and degeneration of cerebral cortex. So that results, since basal nuclei are deteriorated, there's no more proper inhibition of unnecessary movements, so that leads to hyperkinesia and ultimately death. Now, Creutzfeldt-Jacobs disease is quite fascinating. It's a prion disease. Proteins called prions normally exist in our brain, but when a normal prion interacts with disease causing the abnormal prion, the normal one is converted into a bad one. You can see it here in this chain reaction illustration. So normal and diseased interact and form two diseased prions. So that when this pathogenic disease-causing prions accumulate, that results in so-called spongiform encephalopathy. The brain looks like a sponge. The Creutzfeldt-Jacobs disease can be acquired through the consumption of brain tissue or consumption of meat that contains prions. It results in progressive deterioration of the brain tissue and impairment of thinking, depression, insomnia. I have listed all the different changes. So basically, brain is dying. This condition is fatal. There is no treatment. There's no cure. So that'll be it for memory and brain pathologies.